Okay, what I want to do in this video is I want to talk a little bit about your project, Project 3. So some introductory comments. Basically, the goal of this is to use some of the concepts from statistics, as well as some from matrices and linear algebra, and apply this to a real data set. So first let me talk a little bit about the data set. This is a data set that we took in my lab 2.5 years ago. Basically, we had two groups of people, Group A and Group C. And just for some background, we were studying autism. So the individuals in Group A were an autistic group, and the individuals in Group C were a control group. They were ranged in age from 18 to 25. And in this particular data set, what we were interested in looking at is the individuals were given a task that involved using both the auditory and visual system. And we recorded their EEG, or electroencephalogram, while they were doing this task. Then we also put them in the MRI system, and we took something known as a DTI, or diffusion tensor image. So what the diffusion tensor image does is it gives you an image that focuses on connection between different brain regions, and those connections are made by white matter tracks. So when you look at the diffusion tensor imaging data, the data that we're presenting is something called fractional anisotropy. And what that gives you is a number between 0 and 1 that's representative of how intact those connections are between brain regions. Now when you take an EEG, there's a measure known as coherence, EEG coherence. And that is sometimes used to indicate when different brain regions are working have synchronous activity, with synchronous being defined in the electrical engineering way. So in other words, they're basically communicating between each other because of the synchronous activity. So the goal of looking at EEG coherence, it's a measure of what we call functional connectivity. In other words, if you could envision a communication system, the wiring from point A to point B is a structural connectivity. It actually provides the physical backbone for the connections between 2 points. Whereas the integrity of the signal as it's transmitted and whether or not it's correctly encoded and decoded, etc., is a measure of what we might call functional connectivity. So in the brain, EEG coherence measures functional connectivity, and DTI, or diffusion tensor imaging, measures structural connectivity. So the goal of this project is for you to be able to apply some concepts to an actual data set, two different types of data taken across multiple channels. So we have EEG coherence pairs across multiple channels, and then we have DTI measures across multiple white matter tracts. So what we really want to know is there any relationship between this functional EEG measure and the structural DTI measure, and are those relationships statistically quantifiable? So first of all, let's actually look at the data set. So here's the data set. It's given on this Excel file. Our subject group A has group members 1 through 11, and our subject group C has group members 1 through 10. So we have 11 autistic, let's see, are we missing anybody? It looks like we're all intact. We have 11 autistic individuals and we have 10 controls. And so these first two groups are EEG coherence. And then you can see FP1, F7, FP2, F8. These are different channels of the EEG and the coherence between them. So like FP1, F7 is the coherence between channel FP1 and channel F7, and so on. And we're not going to use all of these. This is just the complete data set given in this Excel file. So this is subject group A and subject group C. Then if you look here, these are the DTI, or diffusion tensor imaging data. And you may have to click on the actual location of the cell to see, but for example, the corticospinal tract, there is a right and a left, and so each individual has a number that corresponds with the integrity of the right corticospinal tract and then of the left corticospinal tract and so on. And we've separated out once again autistics and controls. And the reason they're separated out is that we're comparing the autistic group with the control group. So that's your data file. Now, if you look at what the assignment is asking you to look at, is it's asking you to look at these 4 coherence pairs, and those happen to correspond in this data sheet to the first 4 columns of data. So what you're being asked to look at is only the first 4 columns of data on the EEG. The DTI asks you to look at frontal occipital fasciculus left and right and uncinate fasciculus left and right. So if you come to the Excel spreadsheet, that's 4 columns there. And if I'm remembering correctly, yeah, so here is the uncinate fasciculus, and here is the superior frontal occipital fasciculus. So that would be actually columns AR, AS, AT, and AU. So you've got these 4 columns in diffusion tensor image, and then you've got these 4 columns in EEG coherence. And those are the 8 different data variables that we're going to be looking at. So a little word before going through the actual project assignment. It's recommended, I recommend that you use either Excel or MATLAB. If you use Excel, you will have to download the statistical package add-on. That's a little easier if you're using a PC than if you're using a Mac. They're a little bit different. But either way, whether you use MATLAB or Excel, these are standard commands within a standard statistical package that you're being asked to use. So you're not going to have to code anything. You certainly can if you want. You should just be able to use commands directly. Most of the terminology in the project you should have been exposed to in the lectures, but we'll go through very briefly the assignment one step at a time so that we're sure that you know that. If you use Excel, you must attach a copy of your spreadsheet embedded in your Word file. If you use MATLAB, you're going to need to attach a screenshot of your workspace and an M file plus your output. Any figures or tables need to be embedded in the context of a PDF or a Word file. Those are the only 2 forms that I can open. There are some other forms that people sometimes use from Chrome. A lot of times I can't open those. So PDF or Word file, you can put your code or your spreadsheet as an appendix. Some people will use SPSS or R. If any of you have a background in that, you're more than welcome to use that as well. So you have 2 groups, 8 variables per group. So the first thing that you want to do is for each variable and each group, you want to calculate sort of the basic statistical measures. So you're looking for the mean, the standard deviation, the variance, the mode. So first of all, let me suggest, especially when looking at the mode, you're going to need to probably reduce the number of significant digits. Otherwise, you'll never be able to find a mode. You may not be able to anyway. But certainly taking the number of significant digits for calculation 1D, taking that down to 1 or 2 significant digits will greatly change what you can and cannot report as your mode. On the mean, standard deviation, and variance, I would just leave it as is. But even reducing down to 3 significant digits could suffice for the entire project except for 1D where you're going to have to go lower. Then you need to report these variables, and they need to be done in a way that's easy to see. So you do not have to use this table. I've just given you this as an example. You would have, your group A mean, your group C mean, your group A mode, your group C mode, group A variance, group C variance, and then you could plot the horizontal table variables as your different FP1, F7, FP2, F8, and so on. So creating a table like this is a super easy way to present the data. Also possible to present that in the form of a graph where you plot your A mean and your C mean, and then you let your A variables or your 8 variables be your x-axis and your means be your y-axis. If you do that, realize that mean, standard deviation, variance, and mode might be orders of magnitude different, so you probably want a different graph for these four parameters that you're calculating. So we want to do that first so that you can sort of look and see, does it look like there are differences between group A and group C just from an inspection point of view. The next thing we're going to do is we're going to look experimentally at sort of Gaussianity and normality. I've down selected for part A. This is not the case for B and C, but for part A, I want you to plot a histogram. I only want you to do that for 2 of the variables for each group, so you'll actually have 4 plots here. The reason to do that is so that you can look at that and get a visual image for, how close to Gaussian this just appears to be. So this is just kind of a qualitative measure of Gaussianity. Now part B, determine the kurtosis and skew for each variable and group. So what I want to do is pull up MATLAB's help file here. This is some information on kurtosis straight from MATLAB. So if you recall from the lecture that you watched, kurtosis is a measure of sort of how outlier prone a distribution is. So if you have a perfectly normal distribution, your kurtosis will have a value of 3. If you have more outliers than a normal distribution, your kurtosis is greater than 3. If you have fewer outlines, your kurtosis is less than 3. So kurtosis gives you an indication of whether or not you would tend to have outliers in your data set. Skew gives you an indication of how symmetric you are. So if the distribution, if you could visualize the distribution leaning to the left or leaning to the right, that's a measure of skew. Measuring skew and kurtosis give you an indication of how close to normal your distribution is. Because remember, when we do statistical tests, we're making an assumption of normality for some of the statistical tests that we do. So after you determine your kurtosis and skew, then you need to make an assessment as to whether or not the data appear to have a normal or Gaussian distribution. And then you need to assess why or why not. So for example, FP1, FP7 might look like a normal distribution in the control group, and it might not look normal in the autistic group because the skew is too high. So that would be a reasonable explanation and answer. It does not mean you've done something wrong if your data don't look normal. So you shouldn't think that it's supposed to look normal, because in the real world, data sets do not automatically appear normal or Gaussian. Okay, in part 3, I want you to do a t-test between the control group and the autistic group, and then determine if there are any that are statistically different. Okay, this is extremely important. So let's pull up this Excel spreadsheet. Let's look at an example. Let's look at the example FP1, FP7. The t-test is done between the autistic group and the control group. Okay, so you've got these 11 numbers and these 10 numbers, and you're comparing them using a t-test to see if they are statistically significantly different. You will then repeat the procedure for the other variables. So you're actually running 8 separate t-tests between group A and group C for all 8 variables, the 4 EEG variables and the 4 DTI variables. Because what we're trying to find out is subject group A and subject group C different? And so remember, if you fail the t-test and have a p-value of less than 0.05, you can start to make some claims that your data sets may be statistically significantly different. Okay, so we're going to do that. Now, after you do that, I want you to do an assessment for outliers, and I want you to look at outliers with 2 different methods. First, the quartile method, which is described in your book and in the video lectures. And secondly, use a criteria of 2 standard deviations. So what does that mean? Okay, that means if we come to our spreadsheet here, and we calculate a mean and we calculate a standard deviation, if there are any of these numbers that are farther from the mean than 2 standard deviations, we can call that an outlier. And I want you to do this on one variable at a time. So first of all, FP1, F7. look and see if there are any of these subjects that look like an outlier. If there are, okay, then what does that mean? That means that your t-test may not be completely accurate. And so it may be that if you really wanted to do an accurate statistical analysis, you would need to remove the outlier. In the past, I've asked students to do this in the project, and this particular semester, I'm actually not asking you to do that. I just want to know if you can identify outliers. So if you look then at correlation and linear relationships between data sets. So once again, this is done separately for Group A and Group C. All of that with the exception of Part D. Part D, we are going to look at Group C only. So everything else is done for Group A and C. So the first thing we want to do is find the covariance matrix for EEG variables. And then you can find the eigenvalues and eigenvectors for this matrix. And then secondly, we want to find a correlation matrix between the EEG variables and the DTI variables. So in the correlation matrix, it's square, and you'll have on one axis, FP1, these 4 EEG variables. So like you would have row, 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 row. And then your columns, and of course you could do this the opposite way, vice versa. Your columns would be the DTI variables, column, 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 column. And so like row 1, column 1 will be the correlation, the correlation between column 1 variables and row 1 variables. So it's going to be a symmetric matrix, and you're going to expect the diagonals to be 1 because it's the correlation of a variable with itself. So that's what I'm asking for in Part C. Okay, finally, if we look at Part D, pick the 2 strongest correlations from Group C. Plot a linear regression curve with the DTI variable being the independent variable and the EEG variable being the dependent variable. Get the equation for the linear relationship. This is also very easy to do on Excel. In fact, it's probably much easier to do on Excel than on MATLAB because all you have to do is create a plot, and then you go into your plot command and you ask it to do the linear regression, and it'll actually give you an equation for a line. So you can get the equation for the linear relationship. So you're basically picking the 2 strongest correlations. So you'll have 2 plots, one for each correlation. So you're looking at that correlation matrix. You're finding the values that are off diagonal that are closest to 1. And then you're going to plot the regression curve for those 2. And you're only going to do this for the control group. One of the things that I discovered when I analyzed these data was that we could look at these differences between variables 1 and so on using a t-test, and we might find that we don't have anything, and it depends on which variables you're looking at, but we might find that we don't have anything that has any statistical significant difference between autistics and controls. And so it can get a little discouraging because you're looking for ways to distinguish and assist in diagnosis. So when I carried the analysis a little bit further and I started looking at these linear regressions between certain DTI variables and certain EEG coherence variables, what I found was even though the variables themselves might not be different, the regression relationship was quite different. So you would get a very different characteristics of the line for the autistic group than for the control group. So if you want to do more and actually plot correlations for the autistic group as well, plot regressions for the autistic group as well, then that might be some very interesting results that you could look at if you find this to be something you're interested in. But I'm not asking you to do that. So you will need to, when you present your work, you will need to make it very clear each step what exactly you are presenting. I don't want this in answer to question format. I want this in report format. So I expect an introductory paragraph. I expect tables addressing each question to be in paragraph form, but I do not want, for example, for you to take this assignment document and fill in under mean and fill in under standard deviation. This is not short answer form. This is a project, so it needs to be written almost like a lab report, where you would have introductory paragraphs and then you would embed, and you would have explanatory paragraphs. You then embed your data, and then you have a discussion of what the data look like. You can work in groups of 2 or 3 to do this project, but no more than 2 or 3. And once again, of course, as always, do not share work across groups or across individuals, because that is considered academic dishonesty, and that's an automatic 0. So I do expect some differences, certainly in how the data are presented and in the wording of the conclusions and so on. So that concludes this video.